have a thick layer of peptidoglycan in their cell wall, which retains the crystal violet iodine complex during decolorization. Gram-negative bacteria have a thinner layer of peptidoglycan and an outer membrane containing lipopolysaccharides, which is disrupted by the decolorizing agent, allowing the dye to be washed out and the counterstain to be absorbed. Retention of stain. The retention of the primary stain, crystal violet iodine complex, is crucial. Gram-positive bacteria have a higher peptidoglycan content in their cell wall, which traps the dye and resists decolorization. In contrast, gram-negative bacteria have a thinner peptidoglycan layer and are more susceptible to losing the dye during the decolorization step. Diagnostic utility. Gram staining is valuable in clinical microbiology for quickly categorizing bacteria into gram-positive or gram-negative groups. This classification provides initial clues about bacterial morphology, behavior, and response to treatments, aiding in diagnosis and treatment decisions. Limitations. While gram staining is a fundamental technique, not all bacteria fit neatly into these categories. Some bacteria may exhibit variable staining reactions or may not stain well due to their unique cell wall compositions or environmental factors. In summary, the principle of gram staining hinges on the differential retention of crystal violet iodine complex by bacterial cell walls, highlighting the structural differences between gram-positive and gram-negative bacteria. This technique remains indispensable in microbiology laboratories for initial bacterial identification and classification. Importance and applications. ChatGPT. Gram staining is critically important in microbiology due to its numerous applications, which include importance. Bacterial classification. It allows rapid differentiation of bacteria into two main groups, gram-positive and gram-negative, based on their cell wall structure. This initial classification is fundamental for understanding bacterial morphology and physiology. Clinical diagnosis. In medical microbiology, gram staining is used for preliminary identification of bacteria causing infections. This helps healthcare providers choose appropriate antibiotics. As gram-positive and gram-negative bacteria often respond differently to treatment. Quality control. Gram staining is used in quality control processes across various industries, including pharmaceuticals, food production, and environmental monitoring. It ensures the presence or absence of specific bacteria that may affect product safety or quality. Research tool. In research, gram staining provides insights into bacterial behavior, interactions with host cells, and responses to environmental changes. It aids in studying bacterial pathogenesis, antibiotic resistance mechanisms, and evolutionary biology. Educational tool. It is an essential technique taught in microbiology courses to familiarize students with bacterial morphology, staining techniques, and the principles of bacterial classification. Applications. Medical microbiology. Diagnostic tool. Helps in rapid identification of bacterial infections based on gram reaction, guiding treatment decisions. Epidemiology. Enables tracking and studying outbreaks of bacterial diseases by identifying the causative agents. Epidemiology. Enables tracking and studying outbreaks of bacterial diseases by identifying the causative agents. Pharmaceutical industry. Quality assurance. Ensures products are free from harmful bacteria, maintaining safety and efficacy of pharmaceuticals. Food microbiology. Quality control. Monitors food production processes for bacterial contaminants, ensuring food safety and extending shelf life. Environmental monitoring. Water and soil testing. Detects bacterial contamination in environmental samples, aiding in pollution control and environmental health assessments. Veterinary medicine. Animal health. Helps diagnose bacterial infections in animals, guiding veterinary treatment strategies. Industrial microbiology. Biotechnology. Facilitates screening and selection of bacterial strains for industrial processes, such as fermentation and bioremediation. Educational purposes. Laboratory training. Teaches students fundamental microbiological techniques and enhances understanding of bacterial diversity and behavior. Summary. Gram staining plays a pivotal role across various sectors of microbiology and beyond, from clinical diagnosis to industrial applications and research endeavors. Its ability to provide rapid, visual differentiation of bacteria based on cell wall characteristics makes it an indispensable tool for microbiologists worldwide. Examples of gram-positive and negative bacteria and what is the difference? ChatGPT. Gram-positive and gram-negative bacteria are distinguished primarily by the structure of their cell walls, which react differently to the gram staining procedure. Here are examples of each and the key differences. Gram-positive bacteria, Staphylococcus aureus. Found on skin and mucous membranes. Causes skin infections, pneumonia, and food poisoning. Thick peptidoglycan layer in the cell wall retains crystal violet stain, appearing purple-violet after gram staining. Bacillus subtilis. Soil bacterium commonly used in genetic and biochemical research. Produces endospores under stress conditions. Gram-positive due to a thick peptidoglycan layer. Gram-negative bacteria. Escherichia coli. Found in the intestines of humans and animals. Can cause gastrointestinal infections. Thin peptidoglycan layer surrounded by an outer membrane containing lipopolysaccharides, LPS, which are responsible for gram-negative staining, appearing pink-red after counterstaining. Pseudomonas aeruginosa. Opportunistic pathogen associated with hospital-acquired infections. Resistant to many antibiotics due to its impermeable outer membrane. Gram-negative due to the presence of an outer membrane with LPS. Key differences. Cell wall composition. Gram-positive bacteria have a thick layer of peptidoglycan 20 to 80 nanometers in their cell wall, which retains the crystal violet iodine complex during gram staining, imparting a purple-violet color. In contrast, gram-negative bacteria have a thinner peptidoglycan layer, 1 to 3 nanometers, and an outer membrane composed of lipopolysaccharides, LPS, and proteins, which is permeable to the decolorizing agent and counterstain, resulting in a pink-red color. Susceptibility to antibiotics. Gram-positive bacteria are generally more susceptible to antibiotics that target cell wall synthesis, e.g., penicillin, whereas gram-negative bacteria are often more resistant due to the protective outer membrane and efflux pumps that can expel antibiotics. Pathogenicity. Both gram-positive and gram-negative. Susceptibility to antibiotics. Gram-positive bacteria are generally more susceptible to antibiotics that target cell wall synthesis, e.g., penicillin, whereas gram-negative bacteria are often more resistant due to the protective outer membrane and efflux pumps that can expel antibiotics. Pathogenicity. Both gram-positive and gram-negative bacteria include Gram-negative due to the presence of an outer membrane with LPS. Key differences. Cell wall composition. Gram-positive bacteria have a thick layer of peptidoglycan 20 to 80 nanometers in their cell wall, which retains the crystal violet iodine complex during Gram staining, imparting a purple-violet color. In contrast, Gram-negative bacteria have a cell wall composition. Gram-positive bacteria have a thick layer of peptidoglycan 20 to 80 nanometers in their cell wall, which retains the crystal violet iodine complex during Gram staining, imparting a purple-violet color. Cell wall composition. Gram-positive bacteria have a thick layer of peptidoglycan 20 to 80 nanometers in their cell wall, which retains the crystal violet iodine complex during Gram staining, imparting a purple-violet color. In contrast, Gram-negative bacteria have a thinner peptidoglycan layer, 1 to 3 nanometers, and an outer membrane composed of lipopolysaccharides, LPS, and proteins, which is permeable to the decolorizing agent and counterstain, resulting in a pink-red color. Susceptibility to antibiotics. Gram-positive bacteria are generally more. In contrast, Gram-negative bacteria have a thinner peptidoglycan layer, 1 to 3 nanometers, and an outer membrane composed of lipopolysaccharides, LPS, and proteins, which is permeable to the decolorizing agent and counterstain, resulting in a pink-red color. Susceptibility to antibiotics. Gram-positive bacteria are generally more susceptible to antibiotics that target cell wall synthesis, e.g., penicillin, whereas Gram-negative bacteria are often more resistant due to the protective outer membrane and efflux pumps that can expel antibiotics. Pathogenicity. Both Gram-positive and Gram-negative bacteria include pathogens that cause a range of infections in humans and animals. Understanding their Gram reaction helps in selecting appropriate treatment strategies and understanding bacterial virulence factors. In summary, Gram staining remains a foundational technique in microbiology for quickly classifying bacteria based on their cell wall characteristics. This classification informs diagnostic, therapeutic, and research efforts across various fields of microbiology and medicine. Histochemical test for carbohydrates, periodic acid, and CYP reagent method with control and test slides. ChatGPT. Performing histochemical tests for carbohydrates using periodic acid Schiff-Pop reagent involves staining tissue sections to detect the presence of polysaccharides like glycogen, glycoproteins, and mucopolysaccharides. Here's how you can conduct this test with control and test slides. Materials. Tissue sections, control and test samples, periodic acid solution, 1%. Schiff reagent, Schiff's reagent or Schiff's reagent with basic Fuchsin, hematoxylin counterstain. Distilled water. Microscope slides and cover slips microscope. Procedure. Preparation of tissue sections, sectioning. Prepare tissue sections about 5 to 10 microM thick from both control and test samples. Ensure the sections are of uniform thickness to ensure consistent staining results. Mounting. Mount the tissue sections onto clean, labeled microscope slides. Ensure the sections adhere well to the slides without folds or wrinkles. Histochemical staining procedure. Deparaffinization and hydration. If using paraffin-embedded sections, deparaffinize the slides through xylene and grated ethanol series, followed by hydration in distilled water. Periodic acid treatment. Flood both control and test slides with 1% periodic acid solution. Periodic acid oxidizes the diol functional groups of carbohydrates to form aldehydes, which are essential for subsequent staining. Rinsing. Rinse the slides thoroughly with distilled water to remove excess periodic acid. Schiff reagent staining. Apply Schiff reagent, Schiff's reagent with basic Fuchsin to both control and test slides. Allow the reagent to react with the aldehydes formed by periodic acid oxidation. This results in a magenta-purple color where carbohydrates are present. Washing. Rinse the slides gently with running tap water to remove excess Schiff reagent. Counterstaining. Optionally, counterstain the slides with hematoxylin for 1 to 2 minutes to stain nuclei. This step is not always necessary for carbohydrate detection but provides contrast for better visualization. Dehydration and mounting. Dehydrate the slides through grated ethanol series 70%, 95%, 100%, and clear in xylene. Mount the slides with mounting medium and cover with cover slips. Microscopic examination. Observation. Examine both control and test slides under a light microscope using bright field illumination. Carbohydrates will appear as magenta-purple staining against a background. Nuclei may appear blue from hematoxylin counterstain if used. Control slide. The control slide should show expected staining patterns for tissues known to contain carbohydrates positive control such as glycogen-rich liver tissue or mucopolysaccharide-rich cartilage. Test slide. Compare the staining intensity and distribution in the test slide with the control slide. This helps in assessing the presence and distribution of carbohydrates in the tissue of interest. Notes. Positive control. Ensure a positive control slide is included to validate the staining procedure and confirm the reagent's activity. Interpretation. Positive staining indicates the presence of carbohydrates within tissue structures, providing insights into their localization and abundance. Variations. Adjust staining times and reagent concentrations based on tissue type and specific experimental conditions to optimize staining results. By following this periodic acid Schiff-Pop reagent method with control and test slides, researchers can effectively detect and visualize carbohydrates in tissue sections, contributing to studies in histology, pathology, and biomedical research. Using salivary amylase here ChatGPT. If you're interested in using salivary amylase in a histochemical context, particularly in demonstrating its activity through a staining method, here's how you can approach it. Materials. Tissue sections, control and test samples, salivary amylase solution. Staining kit for amylase activity, e.g., amylase staining kit, microscope slides and cover slips, distilled water. Microscope procedure. Preparation of tissue sections, sectioning. Prepare tissue sections about 5 to 10 microM thick from both control negative control without salivary amylase and test with histochemical test for proteins by mercuric grophenol blue method ChatGPT. The histochemical test for proteins using the mercuric grophenol blue method is a widely used technique to detect and visualize the presence of proteins in tissue sections. Here's a detailed procedure for conducting this test. Materials. Tissue sections, control and test samples, mercuric grophenol blue solution, distilled water. Microscope slides and cover slip, sodium carbonate buffer, pH 10 microscope. Procedure. Preparation of tissue sections, sectioning. Prepare tissue sections about 5 to 10 microM thick from both control negative control without protein staining and test with protein staining samples. Ensure uniform thickness and quality of sections. Mounting. Mount the tissue sections onto clean, labeled microscope slides. Ensure the sections adhere well to the slides without folds or wrinkles. Histochemical staining for protein. Preparation of mercuric grophenol blue solution. Prepare the mercuric grophenol blue solution according to the manufacturer's instructions or established protocol. Typically, this involves dissolving mercuric chloride and grophenol blue in distilled water. Treatment with mercuric grophenol blue. Flood the test tissue sections with mercuric grophenol blue solution. Ensure complete coverage of the tissue surface. Incubation period. Incubate the slides in a humid chamber at room temperature for a specified time, e.g., 30 minutes to 1 hour, to allow the mercuric prophenol blue solution to react with proteins in the tissue sections. Washing. Rinse the slides gently with distilled water to remove excess mercuric prophenol blue solution. Differentiation with sodium carbonate buffer. Differentiate the slides by dipping them in sodium carbonate buffer pH 10. This step helps to enhance the color contrast and remove background staining. Counterstaining optional. Optionally, counterstain the slides with a suitable counterstain, e.g., nuclear fast red for contrast, especially if the nuclei need to be visualized along with protein staining. Dehydration and mounting. Dehydrate the slides through graded ethanol series 70%, 95%, 100%, and clear in xylene. Mount the slides with mounting medium and cover with cover slips. Microscopic examination. Observation. Examine the stained slides under a light microscope using bright field illumination. Proteins will appear as blue or blue-purple staining against a background. Nuclei may appear red from counterstain, if used. Control slide. Include a negative control slide where the tissue sections are treated similarly but without the addition of mercuric prophenol blue solution. This helps verify specific staining related to protein detection. Notes. Interpretation. Positive staining indicates the presence and distribution of proteins in the tissue sections. This method can be used to study protein localization in abundance in various tissues and organs. Optimization. Adjust staining times and concentrations of mercuric prophenol blue solution based on preliminary experiments and specific research requirements. The mercuric prophenol blue method for protein detection in histochemistry provides a reliable approach to visualize proteins in tissue sections, aiding in biomedical research, pathology, and histology studies focused on protein expression and localization. Control and test slides ChatGPT. In histochemical staining procedures, control and test slides are essential components used to validate staining techniques and ensure accurate interpretation of results. Here's how they are used in the context of staining for proteins using the mercuric prophenol blue method. Control slide. Negative control. Purpose. The negative control slide is treated similarly to the test slide but without the addition of the staining reagent mercuric prophenol blue solution. Function. It serves as a baseline to assess any non-specific background staining that may occur during the staining process. The absence of staining on the negative control confirms that any staining observed on the test slide is specific to the presence of proteins. Procedure. Prepare tissue sections as usual. Treat the slide with all necessary steps: mounting, incubation, washing, differentiation, except for the application of mercuric prophenol blue solution. Complete the staining process with counterstaining, if applicable, and mounting. Test slide. Experimental slide. Purpose. The test slide is the actual sample where the staining reagent mercuric prophenol blue solution is applied to detect proteins in tissue sections. Function. It provides the experimental data on the presence and distribution of proteins within the tissue samples being studied. Procedure. Prepare tissue sections as usual. Treat the slide with all necessary steps: mounting, incubation with mercuric prophenol blue, washing, differentiation. Complete the staining process with counterstaining, if applicable, and mounting. Importance of control and test slides. Quality control. Control slides ensure that staining procedures are performed correctly and consistently. They help identify any staining artifacts or inconsistencies that could affect the interpretation of experimental results. Validation. Comparing the staining patterns and intensities between the control, negative control, and test slides ensures that any observed staining on the test slides is due to the specific interaction of the staining reagent with proteins, rather than non-specific binding or background artifacts. Standardization. Including control slides as part of experimental protocols helps standardize staining procedures across different experiments and laboratories, improving the reliability and reproducibility of research findings. By carefully preparing and evaluating control and test slides in histochemical staining techniques, researchers can confidently interpret staining results and draw meaningful conclusions about the presence and localization of proteins or other biological molecules within tissue samples. Thymus histology ChatGPT. Thymus histology refers to the microscopic structure and organization of the thymus gland, a primary lymphoid organ crucial for the development and maturation of T lymphocytes T cells. Here's an overview of the histological features of the thymus. Structure of the thymus, capsule. The thymus is surrounded by a thin connective tissue capsule that extends inward, dividing the gland into lobules. Cortex. Outer region. The cortex is the outer part of each thymic. Standardization. Including control slides as part of experimental protocols helps standardize staining procedures across different experiments and laboratories, improving the reliability and reproducibility of research findings. By carefully preparing and evaluating control and test slides in histochemical staining techniques, researchers can confidently interpret staining results and draw meaningful conclusions about the presence and localization of proteins or other biological molecules within tissue samples. Thymus histology ChatGPT. Thymus histology refers to the microscopic structure and organization of the thymus gland, a primary lymphoid organ crucial for the development and maturation of T lymphocytes T cells. Here's an overview of the histological features of the thymus. Structure of the thymus, capsule. The thymus is surrounded by a thin connective tissue capsule that extends inward, dividing the gland into lobules. Cortex. Outer region. The cortex is the outer part of each thymic lobule and appears dark under a microscope. Thymic pastils, corpuscles, concentric whorls of epithelial cells found in the cortex, which are involved in regulatory functions within the thymus. Medulla. Inner region. The medulla is located centrally within each thymic lobule and appears lighter in color compared to the cortex. Thymic medullary pastils, corpuscles, larger and more irregularly shaped compared to those in the cortex, often surrounded by lymphocytes. Cortica medullary junction. The transition zone between the cortex and medulla, where developing T cells migrate from the cortex to the medulla for further maturation and selection. Cell types in thymus histology. Thymic epithelial cells TECs. Found throughout the thymus, TECs play a crucial role in supporting and guiding the maturation of T cells. Cortex. TECs in the cortex contribute to the positive selection of T cells. Medulla. TECs in the medulla contribute to the negative selection of T cells, removing those that may be self-reactive. T lymphocytes T cells. Medulla. TECs in the medulla contribute to the negative selection of T cells. Thymic pastils, corpuscles, concentric whorls of epithelial cells found in the cortex, which are. Thymus histology refers to the microscopic structure and organization of the thymus gland, a primary lymphoid organ crucial for the development and maturation of T lymphocytes T cells. Here's an overview of